So today I'm going to discuss how the brain control uh, breathing or respiration. So we're going to talk about different area in the medulla and pons that control respiration. And as you know, respiration is a autonomic function. So we breathe without our being conscious of doing that. So it's happening spontaneously, even during sleep. But we can control breathing if we need. And so when we talk, so we like adjust the breathing so that we can produce sound. So the main brain regions that are responsible for controlling respiration are the pons, medulla, and there are, of course, other areas that give input to this central controller so they can modulate breathing. So this system has sensor and effector. So it receives input from, for example, the pH of the blood, the oxygenation of the blood. So it's sensed by some sensor. We're going to discuss what are these. And so it receives this input, integrate the input, process it, and generate an output. And so the output is rhythmic breathing, but rhythmic breathing that can be modified or controlled depending on the need of the person, the physiological needs. For example, if you climb a stair, you do exercise, so you need additional oxygen into the blood. So you try to breathe at higher rate. So the effector are the muscle of the in between the rib and the diaphragm. And so as I said, so the breathing is generated by spontaneously active neurons that rhythmically discharge at certain frequency. So when I say spontaneous, it means they do not need any additional input, so they can, it's like the heart, I mean, it keeps beating spontaneously. So there are neurons that generate this rhythmic discharge. So if we stop the discharge of this neuron, so like traumatic injury, for example, to the neck, for example, one way a person can die, for example, if, for example, during trying to jump in a pool and he hit something, so if the neck broken, then he will immediately die because the breathing will stop. So it's a very important region here. You also probably know that one way to sacrifice a mouse is, for example, by separating the neck a little bit or breaking the neck, so the mouse will stop breathing and it will die, immediately. So let me review a little bit the blood circulation and how the lung participate in oxygenating the blood. So you probably have seen this slide before, some of you. So the heart is like a dual pump. So it receives deoxygenated blood. So the darker blood or purple is a deoxygenated blood that contain a higher level of carbon dioxide. And so it pumps this blood, so from the right heart into the lung, and in the lung there is exchange, so CO2 will diffuse out, whereas oxygen diffuse in. So, you know, in the air, there is about 19–20% oxygen and 80% nitrogen, and a very low level of carbon dioxide. So when we exhale carbon dioxide, it's usually at 5% level. And so oxygen will diffuse into the lung in the alveoli. And then, so this oxygenated blood is pumped to the systemic circulation. So, and then it, after it delivers oxygen to the muscle, to the brain, so it comes back to the heart. So here you have the inferior vena cava, and here you have superior vena cava. And so the heart will pump it back again into the lung. Now, the heart is a muscle itself, and it needs oxygenation and blood circulation. So probably have heard about heart attack. It's when the coronary circulation is clogged. So if there is no diffusion of blood and oxygen to the heart, then some of the muscle will die, and so this will cause a heart attack. So the respiratory system anatomy, so it starts with a nasal cavity and oral cavity. So we breathe air through the nose and through the mouth. So that's why during a pandemic like COVID-19, so we need to filter air coming through the nose and the mouth. So when we speak, so also we release air, we release tiny particles that can contain the virus. And so the air enters through the pharynx, then the larynx. And so the trachea is a tubing that contains cartilage, so it doesn't collapse so easily. And then it bifurcates into so many branches. So there are like 5–7 level of branching. So it's like a trees. And it ends in the respiratory bronchiole and the alveoli. And the surface of the alveoli is so big, it's like a soccer field for one person. So it's a huge area. So this branching allow air to diffuse through a big area into the blood, where you have capillary in the alveoli. Now, there are several muscles responsible for breathing movement, and so there are muscles that are involved in inspiration and muscles that are involved in expiration. So the diaphragm is the most important muscle, and it's innervated by the phrenic nerve. So the phrenic nerve are very important to control breathing. So there are internal and external intercostal muscles. So these are the muscles that are in between the ribs. So they help either contract the cage or expand it depending on inspiration and expiration. So as I mentioned, there are several levels where breathing is controlled. So the main generator of rhythm is actually the medulla. And if we sever or lesion at the level below the medulla, we can see that the breathing will completely stop. So any cut below the medulla will prevent this network of neurons from discharging electrical signals that controls breathing. So because they connect the motor neurons that are involved in inspiration and expiration. So if you cut this, you cut the connection, and the breathing will completely stop. Now, what if you make a cut in between the pons and the medulla? You will see that breathing continue, which means it's the medulla that is contain this central pattern generator that control breathing. But at the same time, you see some irregular breathing. So which means that the pons, they somehow modulate the rhythmicity of breathing. So in conclusion, the medulla contain the central pattern generator for generating rhythmicity and controlling the muscle that are involved in cyclic breathing. Now, in addition to the pons and medulla, there are some other inputs that are involved in regulating the breathing pattern. So the vagus nerve, so here you have a column where the vagus or vagi means the two vagus nerve are intact. Here you have the two vagus nerve are cut. Now, what's the role of the vagus nerve? The vagus nerve, or nerve number 10, so it has so many functions, but it also brings sensory information from the diaphragm and the intercostal muscle. And so its effect is to inhibit breathing, or making it higher frequency and shallower. So each breath is take shorter time, but at higher frequency. So when we cut the vagus, we disinhibit, and so the breathing will become increased in depth. So here we cut at different level, and either with the vagus intact or with the vagus cut. And we see if we cut below the spinal cord, I mean, respiration just stop. So if we cut at the level between the pons and the medulla, so we get irregular breathing and an increased like gasping pattern. Now, the pons has two centers, so the apneustic center and the pneumotaxic center. And so these like also cause inhibition of the medulla. So if we cut the inhibitions, then we get more excitation. So slower but higher amplitude breathing. So you can see, for example, if we cut the level between the pneumotaxic center and the apneustic center, we get this apneustic breathing. So when someone so breathes for a long period, it's like a square kind of breathing, and then it releases air. So what are exactly the specific part of the medulla and pons that are involved in breathing or controlling breathing? So in the medulla, we have what we call the medullary respiratory center, and there are two main regions. So the dorsal respiratory group, sometimes we call it DRG, and the ventral respiratory group, or VRG. So these like work antagonistically to control breathing, and they contain so a network of neurons that mutually inhibit each other. But the main neurons that generate like the rhythmic discharge are located on top of the ventral respiratory group. So it's called the pre-Bodzinger complex. Now, in the pons, we have two groups. So we have the one that is the rostral, and it's called the pneumotaxic center, and the caudal part, so it's called the apneustic center. But so the main, so I'm going to give you more details about each group and the type of neuron involved. But the main point of this slide is that the pre-Bodzinger complex have been discovered recently as the main driver of generating a pattern, a rhythmic pattern to control breathing. So now what are the function of each type of each group? So the respiratory center, the dorsal respiratory group, so this one, the more neuron they fire, the more is the depth of inhalation. So if we stimulate this region, the person will inhale deeper, more air. And but so if we inhale a lot of air, then the breathing rate will slow down. So it's like more. There's each cycle there is a bigger exchange of air, but the frequency of breathing is lower. Now the expiratory center, so it's located in the ventral respiratory group. So so the main point here is that these two are antagonistic. One is for inspiration, the dorsal respiratory group, and the ventral group is for expiration. And if we stimulate here, or if we like voluntarily stimulate the ventral respiratory group, like for example, if you want to blow a balloon, so you have to force your expiration. So this means the VRG will be involved, and they will fire more to force the expiration. Now the pons. So we have the upper pons and the lower pons. So the upper pons, it sends continual inhibitory impulses to inspiration center in the medulla. So that's why if we make a cut here, so we have increased depth of inspiration. So this like it's an inhibitory control. It prevents breathing from going too deep. So the function is to make breathing at higher frequency and shallower. Now, the apneustic center located at the lower pons, so stimulation causes apnosis. So it integrates inspiratory cutoff information. Now, what is apnosis? So it's apnosis when you breathe very deeply. So for a longer period, like, and then you release, but don't release all the air. So it happened with us most of the times during the day to like breathe and then release the air. So this is a schematic that show the different input and that control the respiratory pattern generation. So you have in the medulla the ventral respiratory group, dorsal respiratory group, and so we have in the pons the pneumotaxic center and the apneustic center. So now there is input from the vagus nerve, so number 10. So the stretched receptor of the lung. So as I said, if you cut the vagus nerve, you can see that you will get like a change, a modification in the depthness of the breathing. There's also innervation from chemoreceptor and baroreceptors. So there is continuous input to tell the respiratory center what's the level of oxygen into the blood, what's the pH, and what is the pressure, the blood pressure, so that the respiration is adjusted accordingly. Now, the phrenic nerve are the most important to send output to control the diaphragm and to contract the intercostal muscle. Now, how did we discover that the pre-Bolzinger complex is the main central pattern generator of activity for breathing? So we did this in rodent by cutting so brain slices from the medulla. So as I mentioned before, so this nucleus is located at the rostral part of the ventral respiratory group. And you can see it's located also between the nucleus ambiguus and the lateral nucleus, reticular nucleus, lateral reticular nucleus. And here you have the inferior olive. So it's not like a neuron that can be identified in terms of boundary. It's like scatter type of neurons that are interconnected. And so if we do recording from these neurons, we can see a pacemaker activity. And so it's in form of bursting. So you have a generation of burst and pause between the burst. So this, of course, in a brain slice, it's much slower than in the physiological condition. So the brain slice is maintained usually at lower than physiological temperature and in a recording chamber perfused by artificial cerebrospinal fluid. Now, some chemicals that control the rate of breathing is substance P, so stimulate breathing rate. So substance P is excitatory, it's a peptide. Now, some chemicals that inhibit respiration are opioid. So you probably know that opioid overdose. So when someone take opioid for a longer period, and so it like cause inhibition everywhere into the brain, and also cause inhibition of neuron in the medulla, and this will slow down the breathing. Now, you probably, if you are watching George Floyd case, now the defense was arguing that he has some opioid in his system, I think fentanyl, something, a substance that so it's opioidergic, and so it slowed down breathing rate. So this might have contributed to his death, but of course, I mean, the policeman who was sitting on his neck, of course, he obstructed his breathing for 9 minutes, and this was probably the main cause of death. But so opioid overdose can kill a person because it inhibits generation of activity in the medulla. I think probably Michael Jackson also, he had some anesthetic propofol, a substance that also slowed down his breathing, and so an overdose of this substance can cause respiratory arrest, and so lack of oxygen, ischemia, and death. So, so as I mentioned, the rhythmicity occur mostly in the ventral respiratory group at the top part where there is the pre-Bosninger complex. But these area are like interconnected, so they coordinate activity. So respiration is a cyclic activity. So some neurons are excited, other neurons are inhibited. So if we record from like a preparation where we have both pons and the medulla, and we record at different level, so we can record this rhythmic bursting. So here when you're recording from a nerve, so the activity we're recording is like small spikes that are generated by a bundle of axons. So you can see that there is firing and then pause, firing and pause. So there is a burst originating from several axons. And so here we're recording from the ventral respiratory group. You can see the same pattern of bursting. So we can see that there is synchrony between different nerve and the ventral respiratory group. So here you see the phrenic nerve also firing at the same frequency. So all these areas are synchronous. So which means that the entire brainstem medulla, some of the neurons that are involved in breathing, they are interconnected, so they fire together. Now, one preparation that someone has created is by patent, so in UK. And so this is like, it's not in vivo, it's not in vitro. So it's not a brain slice, it's not an entire in vivo. So it's a working heart brainstem preparation. So basically, you want to keep the heart and the lung together with the brainstem, and then cut the brain out, cut the rest of the body out. So basically to isolate the cardiorespiratory center, including the central control. So from this kind of preparation, you can do patch clamping from the medulla. So record single neurons in the pre-Bosninger complex, record nerve activity, phrenic nerve activity, record the heart activity, the electrocardiogram, and record from, for example, the diaphragm contraction. So this is a preparation that can be used, for example, to apply drug, for example, locally, for example, on the brainstem, and see effect on the lung and heart. So it's quite complicated. It's like a zombie preparation, I call it, so because it has no heart, no brain, the rest of the body is cut away. And why it's complicated? Because you have to keep perfusing the heart with fluid and filter it and provide oxygen, because the thicker the preparations, the more it will suffer from ischemia, lack of oxygen in deeper tissue. So that's why you need to make sure that it's perfused by artificial cerebrospinal fluid that is continuously oxygenated. Now, is there any difference between breathing in neonate compared to adult or young rodent? So now, probably in the other course, so I told you that usually during development, neurons are interconnected mostly by gap junction at early stage, and later there is formation of synapse. So early on, so in a neonate, so the neurons are mostly interconnected without chemical synapse, just by gap junction. And so they generate the same rhythmic breathing. But when they mature age, so postnatal day 15, so you can see if we apply strychnine, which is a glycinergic receptor antagonist, so it blocks glycine receptor. So we can see that there is a disruption in the breathing rate. So inhibition is very important in the medulla, but not in neonate. It's a little bit later in adult, when there is formation of synapse and when neurons start to inhibit each other via interneurons. So here I give you more details about the function of the dorsal respiratory group. So it generates basic respiratory rhythm. The respiratory cycle is repeated 12 to 15 times a minute in human, but you can see, for example, in rodent, the breathing rate in rodent is about 3 per second. So it's like 180 per minute. So it's much faster in rodent. So the smaller the animal, the faster respiratory rate, and also the faster is the heart rate. Also, when rodents sniff odor, so this will increase to higher frequency. Instead of 3 per second, it will go to 5 to 8 per second. So the dorsal respiratory group neurons, they have intrinsic ability to depolarize at a rhythmic rate. So there are some intrinsic membrane property. And this is because they have sodium, calcium channel. So appropriate complement of channel that will allow the membrane to spontaneously depolarize, generate bursts, and then go into hyperpolarization in a pause. So quiet breathing inhalations. So the dorsal respiratory neurons transmit nerve impulses via the phrenic and intercostal nerve to the diaphragm and external intercostal muscle. So when all this muscle contracts, the lung will get filled with air. So basically, you're pushing your diaphragm down so that your thoracic cage will increase in volume, and so you inhale air. So now exhalation is mostly like passive. So you stop inhaling, and so the air will leak out because of lack of negative pressure. So the pneumotaxic area, so this is located in the pons. And so it includes two groups, the pontine respiratory group and the central pattern generator. So it's also known as pontine respiratory group. So it's a network of neurons in the rostral, dorsal, lateral pons. So when you say rostral, up, dorsal mean toward the back, and lateral mean toward the side. So one type of nucleus called the cochlear fused nucleus, and also another is called parabrachial nucleus. So these are also involved probably in taste. You probably have seen that before. So it's considered antagonistic to the apneustic center. So it's like the DRG, VRG, they also are antagonistic. So the pneumotaxic and the apneustic center, they are also antagonize each other. So the pontine respiratory group, it facilitates the transition between inspiration and expiration. So basically, it's like it's a modulation, kind of modulation. So it and regulates the depth and extent of inspiration and the frequency of respiration. So it maintains breathing in a quiet way, so higher frequency and shallow breathing. So the pneumotaxic area, it coordinates the control center of the brainstem, regulates the rate of breathing, regulates the length of inspiration, and avoid overinflation of the lung. So which means, let's say if we breathe at a depth, then so the neuron will fire to prevent too much, to stop the cycle and go into inhalation, exhalation. So you inspire, and some neurons fire more and more to prevent excessive inflation of the lung. So it regulates the depth of breathing. So an increase in pneumotaxic output will lead to shallow, rapid breathing, and a decrease in pneumotaxic output will lead to deep, shallow breathing. Now, what are the factors that affect the breathing rate and the depth of breathing? So there are so many factors. So first, so you have the chemoreceptor. So they sense the level of oxygen and carbon dioxide, and the level of carbon dioxide will lead to a change in pH, and so the acidity is sensed in the carotid body, but also the brainstem itself has capacity of sensing the pH level, and neuron will fire more or less depending on the level of pH. So there are some central chemoreceptor and the peripheral chemoreceptor. So peripheral receptor are located in the carotid body and sinus. And the brain itself can sense carbon dioxide and level of pH. So receptor in the muscle of joint. So you probably notice that whenever you start running, you immediately increase breathing rate, and this is because some sensory information is sent to the respiratory center to increase breathing rate to accommodate so the level of activity, of physical activity. There are stretch receptor also in the lung that negatively affect the respiratory center, and they are irritant. So if you breathe dust, for example, it irritates your lungs, so you start coughing immediately. So so this will affect or modulate the respiratory center. Also, higher brain center we can control breathing. So if you do yoga or any kind of meditation exercise, so you can control your breathing rate. So now the brain also higher center can control respiratory drive. So our emotions, so if you are stressed, so you start breathing rapidly. So anxiety can also be manifested in higher rate of breathing. So if you have pain, if there is a startle reflex, so that will increase breathing rate. So there is voluntary control, so we can consciously change the breathing rhythm. So we can cough or sneeze if there is irritant or if there's inflammation in the lung. So the peripheral input to respiratory center, they originate from chemoreceptor, from pulmonary stretch receptor. So it detects how inflated are the lung. So it protects from overinflation. So we call this the herring-brewer reflex. And irritant receptor, so smoke or dust can trigger coughing, sneezing. Now, chemoreceptor are either central or peripheral. So the central, they are responsive to arterial pressure of carbon dioxide. And so the carbon dioxide level is associated with the pH increase. So the higher the carbon dioxide, the more acidity. And so if there's an increase in pH, then the central chemoreceptor will be activated. The peripheral chemoreceptor, they are responsive to decrease in level of oxygen, and also it increases arterial pressure of carbon dioxide. And so whenever you have a change in carbon dioxide level, you also have a change in proton concentration. Now, where are the peripheral receptors located? They are located in the aortic body and the carotid body. So these are the, you have the aortic arc, and so you have this body, they are sensors, so of oxygen level and pH. And so they connect via the vagus nerve to the brainstem, and so they control the rhythmic activity of the central pattern generator. So both nerve 10, the cranial vagus nerve, and the classopharyngeal nerve, they modulate the discharge of medullary respiratory neurons. So glossopharyngeal nerve 9 and vagus nerve number 10. So what do they sense? They sense the level of carbon dioxide, the level of oxygen, and the level of pH. So this is a aorta, so they can monitor directly the level of this substance in the blood. So this is like the oxygenated blood after coming from the lung. They are sensitive to low oxygen concentration below 60 millimeter mercury. So they sense hypoxia, and so they try to generate activity to restore a level of oxygen to normal level in the blood. Let me skip this slide. So it just shows you how the nerve activity discharge in the carotid body to different level of oxygen. So when the level of oxygen decrease, you have increased discharge. So this means there are some neurons that can detect oxygen level and fire according to the concentration of oxygen. If the oxygen is lower, then they will fire more. So the carbon dioxide effect, actually, it's an indirect effect, and it's by modulating the level of proton or pH level. And the direct effect, it's to stimulate the chemoreceptor, and it will trigger an increase in ventilation more quickly than central chemoreceptor. So, so under which circumstances you have like an increase in carbon dioxide? So I can give you example for you. If you like try to asphyxiate someone or put a bag on their head, of course, the exhaled level of CO2 will increase in the atmosphere, and so this will trigger a hyperventilation. So when there is a high level of carbon dioxide, it will trigger hyperventilation, because it's like the body tries to breathe more to get more oxygen. So the receptor will adapt, mean they will not, it will only affect the receptor, give an effect in the beginning, and then it accommodates. So if the pressure of carbon dioxide is too high, the respiratory center will be inhibited. So this is all at the beginning, so you have increased hyperventilations, and the respiratory center will be inhibited later because of adaptation. So there is direct inhibitory effect of hypoxemia. Hypoxemia means a low level of oxygen in the blood on the respiratory center. So any level that is less than 60 millimeter mercury, it stimulates ventilation. So under what condition we have like hypoxemia? So like if you have like COVID, if you have flu that cause pneumonia, so this means that a large part of the lung are ineffective in getting oxygen from the air due to inflammation. So pneumonia is like either viral or like bacterial infection of the alveoli. You probably heard that during COVID-19 you have the cytokine storm. So when the lot of inflammation cause a thick mucus layer into the lung, and this prevent oxygen from diffusing into the blood. So this will cause hypoxia. That's why you monitor the patient. So you test the oxygen level, and if it fell, the oxygen saturation level fell below, for example, 90%, then you need like ventilation or more oxygen. Now, also, if you go to high altitude, like 10,000 feet, or like the Himalaya, so when you hike into mountains, so the atmospheric pressure is lower, so the oxygen level is much lower in the air compared to when you are near the shore, so at zero altitude. So then the body has to adapt, and you might need some increased ventilation to cope with the low level of oxygen in the air. So the peripheral input to respiratory center, the chemoreceptor, they are very sensitive to a pH level. So a decrease in pH, which means an increase in proton ion concentration. So it occurs when carbon dioxide level increase into the blood. And also when lactic acid accumulates in the blood, so during like heavy exercise, So the muscle generate a lot of lactic acid because they could not get enough oxygen, and this will decrease pH or increase acidity. So a decrease in pH is the most powerful stimulus for respiration. So when oxygen concentration is low, ventilation will increase. Now, which part of the brain really sense the level of pH? So it is two parts, the rostral medulla and the caudal medulla, and it's mostly the ventral surface. So so this is like it's in connection with the cerebrospinal fluid. So they are more in contact with cerebrospinal fluid, so they can sense the level of carbon dioxide or pH. Now, how does CO2 is translated into lower pH? So increase in CO2 will lead to a chemical reaction where CO2 combine with water, and there's an enzyme, I think carbonic, I don't remember the enzyme, but so this enzyme will convert CO2 into bicarbonate, and so a lot of proton is released in the process, and this proton will activate the central chemoreceptor. So the capillary, if there is, they have high level of carbon dioxide. Carbon dioxide is a gas that diffuses into the cerebrospinal fluid, and it's converted into protons. So this proton will activate the neurons. So stimulation of the central chemoreceptor will increase the respiration rate, thus increasing blood pH to hemostatic level. So it tries to normalize the level of pH by increasing breathing rate. So when you increase breathing rate, you try to exhale more the carbon dioxide and get more oxygen. So you lower, so you restore pH to normal level. So an increase in carbon dioxide will lead to increased concentration of protons and decreased pH. This stimulates the chemoreceptor, and this happens when there is excessive exercise, accumulation of lactic acid. If you also hold your breath, for example, if you want to dive into the lake, for example, and so you hold your breath, and so you accumulate a lot of carbon dioxide into the blood, and so this will decrease pH, make it more acidic. So there are also some metabolic disease that can lead to increase in decrease in pH or increase in acidity. So a decrease in pH try to restore the respiratory system to hyperventilate, so that it causes blood pH to be restored to physiological range, which is 7.35. And so you increase ventilation, and this increase pH to normal level. So when pH increase, so it becomes basic. When it decreases, it's acidic. So this is a schematic that summarizes a little bit the lecture about the different chemoreceptor, peripheral and central. So I'll let you review it by yourself. Now, let me move to some disease related to breathing. And So sleep apnea is when during the night, so there are episodes where there is, because of obstruction in the throat or nose, so the person cannot breathe for a period of time, and so this will lead to the person having hypoxia in the brain, and so if long term, this can cause neuron to die, so neurodegeneration, dementia. But if it's not like a big issue, it's still, I mean, during daytime, so people will feel sleepy, and because they could not get good sleep at night because of the repeated episode of apnea. So apnea means when the complete cessation of airflow for at least 10 seconds. Hypopnea is like not as severe as apnea, just reduced airflow by 50%, lasting 10 seconds. Now, so apnea occurring during sleep due to disordered control of breathing rather than control to airflow. So it could be either obstruction, so some mechanical obstruction in the nose, or throat, or it could be something inside the brain stem that's not causing like regular modulation of breathing. So the way it's treated is called by CPAP. So it's like continuous pressure. So this is a mask that's put on the nose, and it apply positive pressure continuously so that it help with obstruction. So it tries to remove the obstruction and push more air into the lung so that the lung will not collapse. So it's just a small increase in pressure to help with breathing, and so this prevent apnea. Now, many people suffer with from snoring, so it could occur in normal people, or like if you are tired, and so it's an involuntary, and only the people around you will notice that. So nobody will notice his or her snoring at night. So this is usually a symptom of obstructive sleep apnea. So there are things you can buy on Amazon. For example, you can like expand a little bit your nose, or like some kind of pillow that can help with snoring. You can do also some surgery if there is some obstruction. So snoring also can cause problem during the day, because usually it impede breathing, and when you impede breathing, I mean there's hypoxia, and so people have hard time focusing or remembering things. So which means that it may cause some neuron in the brain maybe to die due to hypoxia, or some synapse to degenerate. So it's important to fix this problem if it's not severe. And so that's it. Please let me know if you have any question. I believe the exam will be take-home question. But if there's any problem with the slide, if something you don't understand, you can contact me, and I'll be happy to repeat or review this in the next time I see you. So next time I'll give you a lecture on central control of the digestive system. Okay. Thank you very much. I hope Dr. Said is still here to stop the recording. I don't know. Okay. Thank you. Bye-bye.
Thank you.
Thank you.
Dr. Hayat, are you done?